Hello and welcome back as we are on the other side of the flip from looking at protein synthesis. Our next voyage into cell biology is going to be to look at cell respiration. And again, this is going to be done in two videos. And so we're going to kind of look at this in two different settings. We'll look at the first third in this video, and then we'll look at the second two-thirds of the process in the next video. And so with that said, let's go ahead and let's jump into cellular respiration. And we'll start with this look at cellular respiration to try to get you to understand that cell respiration is critical in the production and maintaining of ATP. Right? Adenosine triphosphate. We've spent some time already talking about ATP in chapter 2, and here we are in chapter 3, and now we're going to look at the production of ATP, if you would. The process involved is what we call cellular respiration, and cellular respiration is dependent upon glucose. Right? So glucose is needed in order to even initiate and begin the process of cellular respiration. Now, glucose is so important that we can actually take glucose that we've ingested and digested, and we can store it to later convert it back into glucose to go ahead and generate ATP. And so your fat, all right, your fat, your body can take glucose after it's been processed a little bit and convert it into a glycerol molecule that can then be attached to fatty acids to create triglycerides and other types of lipids. Then, when you need that glucose reserve because you're running low or you're working out or you're dieting and you've cut back on carbs or whatever it might be, your body can then go into that fat reserve, remove those fatty acids, take the glycerol, put it back into this process of cellular respiration, and continue to make ATP with it. The danger is when we go to the opposite extreme of not just cutting out, but eliminating completely. And that is where things like the keto diet becomes very dangerous. And so we'll look at that. And we'll look at why fad diets and extreme diets such as South Beach and keto and Atkins, which are all kind of really similar to one another, are actually really detrimental and not healthy in the least bit. And so we're gonna finish up the video 2 with a look at that and touch on it a little bit as we go through this. So let's go ahead and let's start cranking out. Let me remind you that we're going to be dealing a lot with this process of phosphorylation, right, during our look at cell respiration. Sorry, spelled that wrong. All right, phosphorylation. And if you remember from chapter 2, there are actually 3 types of phosphorylation: photo, phosphorylation, all right, which we don't care about because that's done in plants. But we also have this idea of substrate level phosphorylation. And we have this other thing known as oxidative phosphorylation. Both of these processes, both substrate and oxidative, will be critical in us going through and moving through this process of cell respiration. And I'll be sure to point that out as we go through these various stages. So with that said, there are 3 steps to cell respiration: glycolysis, and then there's a little sub step there, if you would, known as pyruvic breakdown or pyruvate pyruvate transformation. But glycolysis is the main step. And then we have the second step in cell respiration, which is the Krebs cycle. And then we end with chemiosmosis and the electron transport chain. And so this video here is strictly focused on looking at this process of glycolysis. In video two, we'll deal with the Krebs cycle and the electron transport chain. And so let's go ahead and start our look at glycolysis. All right. Glycolysis is all about the breakdown of glucose. And so this is where we see the metabolic breakdown of glucose. And the point of breaking down the glucose is a couplefold. One, it's our first time that we're actually breaking down and making ATP. And so we got to look at where is the ATP coming from that we're producing during glycolysis. But the other critical thing is that we're creating byproduct that is going to be later used to help fuel the Krebs cycle and the electron transport chain. And so byproducts of this process of breaking down glucose is going to be later used in the Krebs cycle and electron transport chain and chemiosmosis. And so we'll look at that. All right. So in glycolysis, right, you see I have some highlights here. So we're going to be using enzymes to convert glucose into its final transformation, that being pyruvic acid or pyruvate. Right? Now, pyruvate is a 3-carbon molecule, and it is going to be used to jumpstart or kickstart the Krebs cycle. And so we'll actually start video 2 with looking at pyruvic acid and how it is functional as within the Krebs cycle. All right. But the purpose of glycolysis is through the use of enzymes to break down glucose and create this byproduct of pyruvic acid that is going to be paramount in fueling the Krebs cycle. At the end of glycolysis, we have produced some very important products. We have produced 4 ATP molecules at the end of this process of glycolysis. We have produced 2 molecules of something that we call NADH. NADH. And we have produced these 2 molecules of pyruvic acid. Right? And we've produced these 2 molecules of pyruvic acid. Now, NADH, just for those that in the alphabet crowd that is out there, NADH stands for nicotinamide adenine dinucleotide. All right, so NADH is nicotinamide adenine dinucleotide. Do you need to know that for the exam? No, but I always have the alphabet crowd usually in class, and they like to know what the letters mean. And so nicotinamide adenine dinucleotide is what NADH stands for. The big thing is that NADH is actually going to be used during the electron transport chain, and so we're not going to really touch that until then. The pyruvic acids are going to be used during the Krebs cycle. The 4 ATPs that are created, we bank. Right? We put that into the ATP bank. But let me draw your attention to let me draw your attention to the fact that in glycolysis, we use 2 ATPs. And as is the case whenever you're dealing with a bank, whenever you borrow something from the bank, you have to pay it back. And so, since we invested 2 ATPs and we produce 4 ATPs, we are actually only net 2 ATPs because you got to pay back those 2 ATPs that you originally used during glycolysis. So, what does glycolysis look like? Well, I'm going to warn you that this is a pretty scary picture, and so bear with me. All right. But this is glycolysis. Now, glycolysis, I told you it was scary. Glycolysis is broken down into uh, into two phases. Right? What I like to refer to as the investment phase and the payoff phase. Right? Investment phase, payoff phase. So this is the first part of glycolysis. This is the second part of glycolysis. All right. So in the first part of glycolysis, we have glucose, which is right here. Right? Here is glucose. And glucose is transformed. The first thing that we do in glycolysis is we take glucose and we add a phosphate group onto it. And we do that through the use of an enzyme called hexokinase. Right? Hexokinase. So hexokinase takes a phosphate group and adds it to glucose. Now, when it does that, it uses energy. We've spent ATP. And remember what we said, we had to invest 2 ATPs. Well, here's one of them. All right? The other thing that you need to be mindful of is, what did we just technically do? We just did substrate level phosphorylation. We just use an enzyme to add a phosphate group onto a molecule. And in this case, we added it onto a molecule of glucose. Now, when we do that, we create a molecule that is called glucose 6 phosphate. Right? Glucose 6 phosphate. The 6 represents the fact that the phosphate group was added onto, wait for it, the 6th carbon. All right. And so if we look here, 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. All right, right there. And so glucose 6 phosphate means that we have a glucose molecule with a phosphate added onto the 6th carbon. Now, glucose 6 phosphate, all right, then gets transformed into another structure. So we take the glucose and we kind of rearrange it a little bit. And when we rearrange it, we create fructose 6 phosphate. So we take glucose and we turn it into another 6-carbon sugar, that being fructose. Right? Don't worry about the enzyme that does that. All right? You don't need to know the phosphoglucose isomerase. Right? That's a minor enzyme. Just know that glucose 6 phosphate is transformed into fructose 6 phosphate. But this you do need to know, because glucose 6 phosphate is then, through substrate level phosphorylation, turned into fructose 1,6 biphosphate. 1 because the phosphate was added to the 1' carbon. 6 because phosphate was added on to the 6' carbon, or the 6th carbon in fructose. And guess what drives that? An enzyme called phosphofructokinase. Phosphofructokinase. That is the enzyme that now adds the second phosphate group onto fructose 6 phosphate. Right? So phosphofructokinase, using ATP, there's the second molecule of ATP that we've had to invest, adds a phosphate group onto the 1' carbon, which is now going to create fructose 1,6 biphosphate, because it's 2 phosphate groups that have been attached. This newly created molecule that originated as glucose is then split into 2 3-carbon molecules. There's one 3-carbon molecule. There is another 3-carbon molecule. All right. These 3-carbon molecules are what are actually, if the body does not need to make any more ATP, we take these 2 3-carbon molecules and we store them as glycerol. Right? We actually create glycerol out of these, which can then be used to go ahead and attach fatty acids, which is stored as lipid or fat within the body. Now, we're going to assume that that's not what is going to happen. We're going to assume that now that we've invested these 2 ATPs and we've created these 2 3-carbon molecules from this glucose that came into the investment phase, we can now worry about the payoff phase. And what you need to know about the payoff phase is each of these 2 3-carbon molecules will power the payoff phase. So we run the payoff phase twice. We run the payoff phase twice. All right? So glycerolaldehyde 3-phosphate comes into the payoff phase. All right? And as it's moving through the payoff phase, look what we do here. We create ATP. ATP created. Right? We continue along. ATP over here is once again created. Now, you don't have to be worried about these enzymes that are driving this on the payoff phase. The important enzymes that you need to be aware of are in the investment phase, the hexokinase and the phosphofructokinase. All right? But the end result of moving through the payoff phase and creating these 2 ATP molecules from one molecule of glycerolaldehyde 3-phosphate is the production of pyruvic acid, or pyruvate. This molecule right here is what will be used in the Krebs cycle. But remember, we run this twice. So here's the first glycerolaldehyde 3-phosphate. Here's the isomer of that, the dihydroacetone phosphate. So now the dihydroxyacetone phosphate now goes through this process. It creates another 2 ATPs and another pyruvic acid. So at the end of this, we've created 4 ATPs, 2 pyruvic acids, right, or pyruvate, right? And if you look right here, we created NADH right there. Right? We create that NADH that is right there. And so if you go back to the previous slide, right, you will remember that we actually created two NADHs, two pyruvic acids, 4 ATPs, but remember, we have to pay the bank back for these 2 ATPs that we used. And so we actually have 2 net ATPs. All right? And that is glycolysis. So glycolysis is all about taking glucose, ending up with 2 pyruvates for each glucose that enters into the glycolysis. And along the way, we're creating some ATP, we're creating some NADH, and we're experiencing substrate level phosphorylation. We're building ATP. And remember what I said during chapter 2, that there's only so much ATP that can be stored. In fact, after about 30 seconds of ATP in reserve, it's just going to be wasted because it's highly unstable because of all of the energy within the bonds holding the phosphate groups together. And so we see negative feedback inhibition kick in. What is negative feedback inhibition? Well, we have this enzyme phosphofructokinase that is driving substrate level phosphorylation to create the fructose 1,6 biphosphate molecule, all right, during the investment phase. Well, once there has been ATP that has been produced within the payoff phase, and we've reached that 30-second max of storage, one of those ATP molecules will come back around and will actually bind to phosphofructokinase. And when it binds to phosphofructokinase, it deactivates the enzyme. So that way we're not wasting so much glucose. We will still go ahead and continue to store excess glucose when we need to, but we try not to even get to that point because storing a whole lot of glucose as fat is not advantageous. It's not very beneficial. So high levels of ATP will go ahead and initiate this negative feedback inhibition of shutting down phosphofructokinase. All right. Once ATP levels decrease, then ATP unattaches from phosphofructokinase, and the process resumes. Now, a couple more here that I want to kind of go over. I do want to show you this. Now, this looks scary. I realize that, and I apologize. Not really, but this shows us, all right, here's glycolysis, right? But what if you don't have any glucose? What if you are running low on glucose, but yet you are running a marathon, or you're lifting and heavy weights, or you're just working out, maybe you're doing a HIIT routine, and you need to have that ATP reserve and or levels at hand so you can go ahead and fuel your muscles to be able to do muscle contractions so you don't cramp up. What if you're out of that glucose, though? What can you do? Well, this is where the body will go ahead and dip into those triglycerides that it's storing as fat, remove that glycerol group, and convert the glycerol back into that glycerolaldehyde 3-phosphate molecule to be able to run the payoff phase. Right? This is what we would define as the formation of glycerol is lipogenesis. All right? But lipolysis would be the breaking down of that fat to be able to create the glycerol that can be converted back into the glycerolaldehyde 3-phosphate to run the payoff phase. Now, the fatty acids will actually go in and can be contributed to the Krebs cycle, which we haven't talked about yet. All right. But there's more to this story because not only can we obtain glucose, right, or glycerolaldehyde 3-phosphate from fats, we can also do it from proteins through the breakdown of proteins into the amino acids and creating something that we call a keto acid. And those keto acids can also then be transformed into the glycerolaldehyde 3-phosphate to fuel the payoff phase. But here's the danger in that. And this is where diets like not South Beach, but well, yeah, South Beach, but what's been rebranded as keto. Here's where the danger comes into that. You're starving your body of carbohydrates, so it has to pull from something else. Well, if you're already low on fat, if you already have a lower body fat percentage, where else is it going to get that protein from? Or where else is it going to get the glycerolaldehyde 3 phosphate from? From proteins. And if you're doing the keto diet, technically you should be doing what? You should be upping the amount of proteins that you have. But what usually ends up happening is your body will burn through that protein very quickly because it's not an efficient means of supplying glycerolaldehyde 3 phosphate. It's not efficient. If it was, your body would do it automatically. And so you've got to way increase that protein to even become near to enough protein to be able to go ahead and break it down to be able to create that glycerolaldehyde 3 phosphate. What typically ends up happening is you never elevate that level of protein to the level that it needs to be at, and you start breaking down your own protein. You start losing body mass. You're not losing fat. You're losing muscle mass. Muscle mass weighs more than fat, which is why people on the keto diet experience such rapid weight loss. They're losing muscle mass. Any diet, any fad that takes you to an extreme to say where you have to eliminate this and you have to supercharge this is not healthy. Your body works in the middle. That's what chapter 1 was all about. That is homeostasis. Your diet is no different. Diet is no different. So, last slide. So what do we end up doing with that pyruvic acid or that pyruvate that we have created? Well, good question. The answer to that is we take the pyruvic acid and we actually join it with another molecule called coenzyme A. And when we take the pyruvic acid and we join it with coenzyme A, we create something called acetyl-CoA. And acetyl-CoA is what will be used to kick off the Krebs cycle so we can continue this process of making some more ATP. Remember, at the end of glycolysis, we have 4 total ATPs that have been produced to net because we have to pay back the investment phase. Right? So during the Krebs cycle, we're going to see some more ATP made, but more importantly, we're going to be seeing some more of those NADHs and something that's called FADH2 produced, which is going to be important for driving the electron transport chain. And so you have a lot to digest in this video. So we're going to stop here. I'm going to let you do that. I'm not even going to try to flip you on this video because that's how dense this content is. I'm just going to push you into the next video. And with that, I'll catch you later.